Welcome.
Thank you, Chang. First of all, I'd like to thank the organizer for such a wonderful meeting and having me here. In the past few years, as a virologist from Wuhan, we put tremendous effort on SARS-CoV-2, but I still think herpesvirus is one of the most successful virus and complicated virus, and also my favorite virus. Today I'm going to talk about the regulation of KSHV life cycle. And the second part, I'm going to share with you a small story regarding a host factor NDRG1 and facilitated both latent and lytic replication. So as you may know, KSHV is the newest discovered herpes virus. This year it is the 30th anniversary of the discovery. And it's a large double-stranded virus. And this virus a little bit smaller than HCMV, just mentioned by Dr. Gondry. In vivo, this virus mostly infects endothelial cell and B cell, cause Kaposi sarcoma with endothelial cell origin, and 2 B cell lymphoma, primary infusion lymphoma and multicentric Castleman disease. So not like other herpes virus, KSHV is not that ubiquitous in human. It only infects 5 to 10% general population. However, in some endemic region like Mediterranean country and African endemic region, Central African, the infection rate can reach to 30 to 50%. In China, there is also an endemic region called Xinjiang, Uyghur Autonomous Region. 14 years ago, a postdoc in my laboratory screened thousands of blood samples from health donor. She confirmed the infection rate in this region is ranging from 20% to 40%. So I think this can explain why the Kaposi sarcoma patient without HIV co-infection can be frequently seen in this region. Yeah, like other herpes virus, especially HCMV, just mentioned by Fleischer, this virus also have 2 modes of life cycles. After primary infection, the virus can quickly establish a latent infection with only subset limited number of genes expressed during latency. However, under some pathological condition like immunosuppression, hypoxia, stress, the virus can be triggered into lytic phase. So a number of groups already showed this RTA encoded by OR50 is a master switch protein can regulate the virus reactivate from latency to lytic replication. Besides, RTA is a potent transcription activator. This protein also can function as E3 ligase, or indirectly binding and degrade proteins by recruiting host E3 ligase. We and others already showed that there are a number of important substrate proteins, like including IRF7, IRF3, which is important in innate immune response. And also HEY1 and KBP1, which is a co-repressor. In this way, RTA can create a favorable environment for the virus undergo lytic reactivation. So on the other side, when I get into this field, I was a postdoc in Dr. Earl Roberts' lab. We found that this nanoprotein is a major latent protein. The major function of this protein is to tether the viral genome to the host chromosome, and through its C-terminus can bind to the terminal repeats of the viral genome, and the N-terminus of this nanoprotein can interact with histone protein. In this way, tether the viral genome to host chromosome. We also found that nano can control the viral gene expression through directly target RTA protein. It can directly interact with each other. In this way, nano can block the RTA autoactivation and its transactivation of its downstream genes. In this way, shut down the viral gene, lytic gene expression to help the maintenance of viral latency. So later on, we also showed that nanoprotein can interact with Cap1, also known as TRIM28. It's a co-repressor. And this protein can then recruit some other epigenetic regulator like HDAC1 and SDDB1. In this way, can modify the histone proteins to silence the viral lytic gene expression during the de novo infection. In this way, help the virus establish latency. So quite interestingly, Zhejiang's group also showed that nano is a major protein which can recruit polycomb repressive complex like PRC1 and PRC2 to the viral chromatin. And in this way, silence the viral gene expression, facilitate the latency establishment. So currently, the field is widely accepted this model. In this model, nano and RTA, these two major viral proteins in charge of regulating latent infection and lytic replication. But the detailed mechanism in this model still need further explore. It's quite interesting. You can see this model is very much fit our Chinese yin-yang and tai chi model. So next, I'm going to share with you a small story about this host protein, NDRG1. So during the latent infection, the KSHV genome persists as a multi-copy circular episome in the nucleus. Typically, one pale cell, if we culture in the cell culture, you can find about 50 to 200 copies of the viral genome within one cell. So KSHV genome replicate once per cell cycle and segregate into daughter cell along with the host chromosome during cell division. So in this simple model, you can see the nanoprotein tether the viral genome to the host chromosome. And very important in viral DNA replication. So as you know, nano doesn't have any enzymatic activity related to DNA replication. It must recruit some other host replication machinery to this replication bubble to finish the DNA replication process. So to understand the mechanism, we also did some omics study, and with integrative analysis, we found this NDRG1 and downstream regulated gene 1 is highly upregulated in KSHV infected cell. So this NDRG1 belongs to the alpha beta hydrolase family, but it is an inactive member of this family and also highly phosphorylated protein. Actually, this protein has been extensively studied. It is involved into a number of important cellular processes, but a very few studies link this protein to virus infection. So if you look at the expression pattern of this protein, you can say it is widely distributed in different tissues. But surprisingly, we found this protein is non-detectable in B lymphocyte, as well as the endothelial cell. These two cell types, as I mentioned to you, is highly permissive for KSHV infection. So this made us to hypothesize the KSHV infection upregulates this protein. It may have some role in KSHV DNA replication. So to test this hypothesis, we confirmed our omic data, and we showed, here we can say that this NDRG1 is strongly expressed in KSHV-infected mesenchymal cell. And we also confirmed this result with 3 pale cell lines, which KSHV latently infected the cell. And compared to the KSHV-negative B lymphoma cell line, you can say it's just 0 to 1 expression of this protein in KSHV latently infected cells. After we lock down the NDRG1 expression, we can see a significant loss of KSHV genome in KSHV infected cell. And we confirmed this result with these 3 KSHV positive cell, and we got a similar result. If we lock down the NDRG1 expression, seems the viral genome within the cell cannot be maintained very well. So we even do a fluorescence in situ hybridization, directly visualize this result. You can see this is a control cell. We have very strong signal of KSH viral genome, and we have very less signal in this lockdown cell. So above result suggests NDRG1 may have some role in DNA replication, but previous study never showed this. So to study the mechanism, we did mass spec experiment for NDRG1. Quite interestingly, we found about 14% of the potential binding partners of NDRG1 is related to the DNA replication. The number 1 protein is this PCNA, proliferating cell nuclear antigen. Yeah, this protein is very important in DNA replication. It can function as a DNA polymerase clamp. With recruitment by RFC protein, it can circulate the DNA and holds the DNA polymerase slide on the double-strand DNA, facilitates the elongation process. So we found this, we confirmed the interaction between NDRG1, and the PCNA. You can see here the NDRG1 and PCNA can directly interact, and it also very nicely colocalized in the cell nucleus. Yeah, we further found this NDRG1 can form a complex with LANA and the PCNA. 3 proteins can nicely colocalize in the cell nucleus. But we also found this LANA protein cannot directly bound to PCNA, but it can interact with NDRG1. In the presence of NDRG1, LANA can nicely pull down PCNA. So we further found this NDRG1 can help LANA loading PCNA onto KSHV genome by using pull-down assay with this TR region. And further, we showed this recruitment by LANA that the PCNA can facilitate the replication of a viral genome. So here, come up with our working model. Is in this model, we showed with the KSHV infection, NDRG1 can highly upregulate it. And simply, it can recruit this DNA replication machinery to the host, to the viral genome and facilitate the viral genome replication, then maintain the viral genome copy number in the KSHV infected cell. So next, we also showed if we lock down NDRG1 expression, the KSHV lytic replication also heavily impaired. But the mechanism is completely different. We showed that NDRG1 can interact with ORF44. So this protein actually is a lytic gene. It's a downstream gene of RTA. It can function as a DNA helicase. If we lock out this ORF44, the lytic replication process can be totally shut down. So to make a long story short, we actually showed this NDRG1 can interact with ORF44. With this interaction, it can enhance the stability of ORF44 during the lytic replication. In this way, facilitate the viral lytic replication. So I think I stop here. I'd like to take the opportunity to acknowledge my collaborator, especially Dr. Kincaid from Harvard, provide us the 8-copy TR plasmids for replication assay. Here is the funding agency and my group. Thank you very much. Take some questions.
Thanks. We have enough time for question. Yeah. Guigan.
Yes, thank you. So I have a very short question. So in the latent stage, do you have any idea how this gene is upregulated? Because you see in latency, this phenotype is very, very strong. The protein expression is very, very high. So do you know how this in latency is upregulated? Thank you.
Yeah, you know, during the latency, there are only several genes get expressed. We checked all of these genes. And I didn't show data here because of time limitation. The LANA protein can upregulate the promoter of this NDRG1. And if we put a stop codon on LANA, this mutant virus cannot upregulate NDRG1. That's what we know now.
Okay, thank you.
That was a really interesting talk. 2 questions. One, KSHV has its own processivity factor, a homolog of PCNA. And so do you know, is there an interaction there, or is one acting in replication of the viral genome for lytic infection versus latent?
Oh, that's very interesting to test. We didn't do that experiment. But during the latency, the KSHV own replication machinery doesn't express. So it only can recruit the host machinery to express.
Right, so is the KSHV processivity factor not expressed at all? You can't detect?
I cannot test.
Yeah, okay.
Yeah.
And then Kenneth K just had a really beautiful nucleic acids research paper out on the conformation of the genome being controlled by host DNA damage pathways. Do you know if NDRG1 has any effect on the conformation of the genome, the circularization for latency?
I have no answers. Thank you.
Okay. If no question.